A koniocellular cell is a neuron with a small cell body that is located in the koniocellular layer of the lateral geniculate nucleus in primates, including humans. Koniocellular layers are located ventral to each parvocellular and magnocellular layer of the LGN, even if the quantity of neurons is approximately equal to the number of magnocellular cells. The koniocellular layers are much thinner due to their size. In comparison to the parvocellular and magnocellular system, fewer studies have been conducted to investigate the koniocellular system. Koniocellular cells are a heterogeneous population differing in many aspects, such as response properties and connectivity, structure. K cells are neurochemically and anatomically distinct from M and P cells. There are three proteins by which K cells can be clearly distinguished: calbindin, the alpha subunit of type II calmodulin-dependent protein kinase, the gamma subunit of protein kinase C. K cells differ in the size from M and P cells. They are much smaller. Unlike M and P cells, K cells are structurally similar to other thalamocortical neurons. This suggests that K cells act like other thalamocortical cells. Function: since K cells are a heterogeneous group of cells, it is likely that they contain subclasses which fulfill different functions. Some cells respond to color, some to achromatic gratings, and still others are unresponsive to any types of gratings. Experimental results suggest that K cells could contribute to aspects of spatial and temporal vision, but it is unclear exactly how. Some hypotheses are: K cells contribute to brightness contrast information and color contrast in species with color vision. K cells contribute to eye movement-related signals by directly projecting to the dorsomedial visual area, which is a motion-related area. K cells are part of a neuromodulatory pathway by projection to the most superficial layer of V1 layer I. Layers ventral to the magnocellular and parvocellular layers lie the koniocellular layers, which differ in thickness. In macaques there are 2 magnocellular and 4 parvocellular layers, and accordingly 6 konicellular layers. K1, the layer ventral to M1, is the largest. K2, K3 and K4 are thinner but nonetheless substantial bands of neurons. The 2 most dorsal layers, K5 and K6, are mostly monolayers. Similar in physiology and connectivity to W cells in cat LGN, K cells form 3 pairs of layers in macaques. The middle pair relays input from short wavelength cones to the cytochrome oxidase blobs of primary visual cortex. The dorsalmost pair relays low acuity visual information to layer I of V1. The ventralmost pair appears closely tied to the function of the superior colliculus. K cells are not restricted to the koniocellular layers. They are also found in small groups, in pairs or as single cells within M and P layers. Larger subpopulations form bridges spanning the distance between 2 adjacent K layers. Input: each koniocellular layer is innervated by the same retina part as the M or P layer dorsal to the respective K layer. Thus, the LGN contains 6 koniocellular layers. K1, K4 and K6 receive contralateral retinal inputs, and K3 and K5 receive ipsilateral retinal input. K2 receives input from both retinae, but the input from the 2 eyes is relayed in separate tiers. The more dorsal tier is innervated by the ipsilateral retina, and the more ventral is innervated by the contralateral retina. K cells receive input from a heterogeneous group of wide-field cells, including small bistratified cells, sparse cells, and possibly also large bistratified cells and broad thorny cells. Those bistratified cells are ganglion cells that send short wavelength signals to the LGN. Retina geniculate axons terminating in the middle K layers display center-only blue on yellow off receptive fields. Sparse cells are presumed to transmit blue-off signals. Both small bistratified cells and sparse cells project to K cells. Therefore, K cells are believed to relay short-wavelength visual information. Cortico-geniculate axons appear to be quantitatively dominant within the LGN. The same holds for K cells, but unlike M and P cells, they also receive input from the extrastriate cortex. Axons arising from the superficial gray layer of the superior colliculus terminate in every K layer, with the most ventral layers receiving the strongest input. Thus, it is assumed that the K layers are functionally related to the superior colliculus, e.g., reflexive control of eye movements. As a conclusion, retinal inputs compete with a quantitatively dominant corticothalamic innervation and a rich innervation from brainstem nuclei. Output: K cells terminate in the superficial blobs and layeri of V1. The dorsal-most K layers have many axons terminating in layeri of V1, whereas K1, K4 rather send their axons to the blobs. However, this division is not clear-cut. For example, it has been found that axons from neurons in the ventral-most pair innervate layeri of V1, 2. The innervation of blobs follows the pattern known from the retina-geniculate terminations. Neurons in layers K1, K4 and K6 terminate in blobs at the centers of the contralateral eye columns. Neurons in layers K3 and K5 terminate in blobs at the centers of the ipsilateral eye columns. Neurons in layer K2 terminate in both. In macaques, about 30 K cells send their axons to one blob. Anatomically distinct subpopulations of K cells innervate different types of blobs, such as blue, yellow blobs or red, green blobs. Neurons in these blobs display blue, yellow antagonism or red, green antagonism. Moreover, K cells innervate extrastriate areas. These K cells are rather large, sending their axons to V2 and inferotemporal cortex. Immunostaining revealed only a few sparse and broadly distributed large K cells. Apart from the K cells innervating the foveal representation of V2, which are more densely packed and found along the caudal and medial margin of the LGN. Throughout each K layer there are neurons that innervate the extrastriate cortex and that are likely to sustain some visual behaviors in the absence of V1. The fact that K cells directly project to HMT supports this hypothesis. Development and plasticity. It is assumed that K cells generate and migrate contemporaneously with neighboring M and P cells. Neurons in the most ventral part of the LGN develop before neurons in more dorsal layers. Neurons of layer K1 develop close to the time of final mitosis for neurons in layer M1, and neurons of K6 develop slightly before neurons of layer P6. While M and P layers in LGN and their axonal terminations in V1 degenerate after a loss of patterned visual input, K cells are not affected. A theory for blindsight. Blindsight is the phenomenon where patients with injury in the primary visual cortex show persistence in motion detection without visual awareness. The brain area responsive to motion in the human brain is called V5 or HMT. Many approaches have been examined to reveal the underlying mechanisms of blindsight. In the past it has been shown that superior colliculus ablation has an effect on V1 independent vision, which in turn advocates the role of the superior colliculus for blindsight. In case of V1 lesions, additional LGN inactivation leads to a strong reduction of neural activity in the extrastriate areas, such as mount. Research has shown that there exists a direct pathway from the LGN to mount consisting mostly of coniocellular cells. In fact, 63% of the neurons directly projecting to mount are coniocellular cells. The input mount receives directly from the LGN makes up about 10% of the V1 neuron population projecting to mount. These results suggest that the coniocellular layers play a key role in V1 independent vision, since the coniocellular layers receive input from the superior colliculus. The previously obtained results can be complemented by the role of the coniocellular layers.